Welcome to the Ontario Aquaculture Research Centre at the University of Guelph. At this facility, we maintain 3 in-house breeding populations of salmonid fishes, and this video will discuss reproduction and spawning. Salmonid fishes include salmon, trout, chars, and freshwater whitefishes. Salmonid fishes are common in aquaculture production worldwide. The Research Centre's broodstock includes a variety of salmonid fishes. In particular, we breed 2 populations of rainbow trout, one that spawns in the fall and one that spawns in the winter. We also breed Arctic char. All of these animals are housed in large outdoor tanks which are exposed to a natural photoperiod. These tanks are 70,000 liters in volume and hold both male and female fish. In temperate fishes, photoperiod is the main factor driving the timing of maturation. Water temperature also has a role to play, where high temperatures may inhibit ovulation and reduce fertility. Because we use a groundwater supply, the water temperature at the Research Centre remains consistent throughout the year, reaching lows of 8.5 degrees Celsius in the winter and highs of 12 degrees Celsius in the summer. In preparation for the spawning season, we use a seine net to capture all of the adult animals in the brood holding tank. These animals will range in age from 3 years to 6 years, and they will range in body weight from approximately 1 kilogram to 8 kilograms. With the help of a tractor, we are able to move the brood into a series of rectangular raceways where the animals are easier to handle. During this step, we are able to sort the animals into groups of males and females by evaluating their secondary sexual characteristics, or how they look. Females tend to be lighter in color and have a round, soft abdomen, whereas males tend to be darker in color and they have a more firm and more triangular shaped abdomen. And sometimes males will develop a dorsal hump or an elongated lower jaw that's called a kype. Once in the raceways, the broodstock are checked weekly for ripeness, which means we're checking for the presence of gametes. This involves using a light sedative to make the animals easier to handle, and the staff will run their hands along the abdomen of each fish. If gametes, either eggs or sperm, which is also known as milt, are free-flowing from the fish, then those animals are considered ripe and they're ready to spawn. The ripe animals are then moved into a holding tank in our spawning room. In groups of 2 or 3 fish at a time, animals are netted and moved into a sedation tank. We use an approved chemical that moves across the gill by diffusion, and it acts as a sedative or a general anesthetic that depresses the sensory centers of the central nervous system. Once the fish are placed into the sedative bath, they should lose equilibrium and start to roll over in about 3 minutes. We know that the fish are sedate enough to be handled if they do not respond to touch. Effective sedation allows us to handle these animals for spawning with no risk to the fish. Females are removed from the sedation tank, they're quickly rinsed in fresh water, and then patted dry with a towel. We use a scanner to check for the PIT tag. This is a passive integrative transponder containing a unique identification code for each animal. Using gravity to assist, females are positioned so their heads are held higher than their tails. A technician then applies pressure to the abdomen of each fish to remove the eggs from her body. This process is also called stripping. Eggs are collected into dry containers, and the technician ensures that they hold the fin away from the vent to prevent the eggs from getting wet. This spawning practice is non-lethal, and it allows for the reconditioning of the fish so it may be spawned again next season. After the eggs are removed from the female, we collect a body weight and a body length measurement before returning the fish to the recovery tank. In general, the size of the egg depends on the size and age of the parent fish. The larger specimens produce more and larger eggs. Sperm, or milt, is collected from the males in the same way as the eggs are collected from the females. Pressure to the abdomen will result in sperm being ejected through the vent where it is collected into a dry container. After measurements are collected on the males, they too are returned to the recovery tank. Once all the gametes are collected from both males and females, we move to the lab where we plan our crosses and fertilize the eggs. We aim for crosses between individual males and females across generations in an attempt to maximize the individuals contributing to the next generation and reduce inbreeding. For example, if we collected eggs from a 4-year-old female, we would use sperm from either a 3 or a 5-year-old male. As our brood population normally has fewer males than females, we often end up reusing some of the males later in the spawning season. Unlike females, males will continue to produce sperm over a period of weeks. Sperm is added to the eggs and stirred gently, followed by a small amount of water to activate the sperm. The containers are left for about a minute to allow the sperm to fertilize the eggs. Once the eggs are fertilized, a little fresh water is added and then the eggs are washed clean. During this process, excess sperm, poor quality eggs, and unfertile or white eggs are removed. Unfertile eggs are white because the proteins coagulate in the eggs, and poor quality or moon-eyed eggs end up floating to the top. This makes these easier to remove. The goal is to remove anything that would cause fungus to grow while the eggs are incubating. After washing, water is removed from the fertilized eggs using a strainer and a towel. We then count out 100 eggs from each cross and we measure the weight of those eggs. We can use this information to estimate the weight of a single egg. The total egg batch is then weighed to get a measurement of the egg mass. We then use the weight of a single egg to estimate the total number of eggs, and that's how we know how many eggs we're stocking into each incubator. The eggs are placed into a stacked incubator tray at 8.5 degrees Celsius, where they will remain until they hatch. The incubation time is temperature dependent, so at this water temperature, rainbow trout eggs will hatch in approximately 45 days. Arctic char eggs, however, are incubated at 5 degrees Celsius, and this results in their hatch in approximately 60 days. We can use the temperature to predict critical time periods like eye up, which is when the development of eyes in the embryo are visible to you. We can also predict the timing of hatch and the timing of first feeding. During the incubation period, it's important to remove any dead eggs or eggs which are developing signs of fungus. This can be accomplished by hand or by using a commercial egg picker. In the case of fungus, we can also accomplish this with a chemical treatment. Fungus will spread really quickly from dead eggs to live ones, and it's important to manage this issue before it becomes a problem so that we can prevent early egg mortality. After hatch, the embryos still have a yolk sac, which they will use as a source of food for approximately 2 weeks. As the embryo consumes more of their yolk, they become more active, and they will soon require an external source of feed. Once the fish are feeding on an external food source, the success rate of these animals is actually quite high. Some families will have a better success rate than others, but generally speaking, about 80% of the rainbow trout eggs that are spawned at the research center will hatch. In some cases, we'll see families that have a higher rate of developmental deformities like scoliosis, and these families are culled from the population as the fish grow. Juvenile fish are reared in the hatchery facility. Some of these animals will end up in research projects, and eventually some will be saved for future broodstock replacement. Each year, the breeding cycle starts over again, and this helps to ensure that we have adequate fish for research projects at the Ontario Aquaculture Research Centre. While the age at maturation, the time of year that spawning occurs, and the incubation temperatures in the hatchery may vary depending on the species that we're talking about, all of the salmonid fishes at the research center have a similar reproductive cycle. So we can apply this practice to all of our fish that we keep at this facility. Thank you for watching, and I hope you enjoyed our video on spawning and reproduction.